very basically, if we look at chromatography, we are separating substances due to their differences in solubility. And just very simply, in its simplest form, a chromatogram is, for example, a piece. I use filter paper because it works well with water as the solvent. I draw a pencil line a small distance from the bottom, okay? And on that pencil line, we can put tiny little samples of, well, for example, ink. Now, yes, but then you need a different solvent. I'm using water as a solvent. It's substances. I'll explain to you in a moment. Okay. The chromatogram, you usually hang it up. You can put a ruler across the top of the container and a little bit of sellotape, and you hang your chromatogram up so that just the lower edge is in the water. We're using water as a solvent, so you can assume that the pigments that we are using are also water soluble. If I use, for example, a permanent Cokie, I won't get a result because the permanent ink has a different solvent. But water as a solvent, many of your inks are water soluble. Those are probably all water soluble. Okay. So what is going to happen is the water is going to be drawn up this chromatogram. But together with the water, as it is drawn up the chromatogram, the pigment in the inks also dissolve in the water. And the water takes it up along the chromatogram. And we find very simply that if the water has reached the edge of the paper, then you stop the process, you take it out of the water. You will find that the different pigments will travel up to different heights. All right? If I had an extra one over here consisting of blue and green and red ink, then I will find the following over here. There, there, and there. Do you follow? Well, it depends on what pigments are used to make the black ink. Different companies use different pigments, okay, or different dyes. So you could do a chromatogram, and this is what they do in forensic science when they, for example, try and figure out which pen wrote the note. They can do a chromatography of the ink, and they can trace the ink. Well, if they've got the particular pen, they can trace it back to a particular pen. Okay. The larger companies with the fountain pens, the more expensive ones, have got specific batch lots that they know what dyes they use, and you can even trace it back to a specific batch lot on when you start investigating these things. Now, when we use colored substances, it's very easy to see. Oh, okay, these two had the same thing, those two contain the same thing, and those two contain the same thing. Can you see that? All right. Now, we can work out a mathematical volume. We talk about an RF value. Now, the RF value is the distance traveled by the substance. Okay, so distance traveled by substance. Okay, yes, divided by the distance traveled by the solvent. Distance traveled by substance, okay, divided by distance traveled by solvent. And then just remember, if I asked you which pigment is more soluble, the red one is more soluble. Why? It traveled up the highest. Okay. Along with the pigments. Now, for example, in medicine, they can test urine samples for sugars or proteins. These sugars and proteins do not have colors. So you're going to get a chromatogram with invisible spots on them. So what they go and do is they treat those invisible spots with something known as a locating agent. All right. Now specific substances require specific locating agents, but we aren't doing medical science now, so just very simply, this looks terrible, okay. A locating agent is just something they will spray on the chromatogram to allow the points to become visible. Do they have to have different colors? No, because if my chromatogram, if I have got one, there's my chromatogram, and there's my pencil line, and I had different spots of whatever substance I had on there, and I got a result like this. What can I deduce from my chromatogram? Well, let's say A, B, C, and D. It doesn't have to have a different color. It must make a visible spot because different substances have different solubilities. Okay. So we can go and figure out substance A and substance C shares one substance. Substance B shares one substance with A and one substance with D. Substance C contains something that none of the others have, and it corresponds with one point on A. All right. And then substance D has the same substance in it as B. Now they can use this for coloring in cool drinks to test which chemicals they've added. We can do it with sugars. We can do it with proteins. You can do a chromatogram with chlorophyll because the chlorophyll in the leaves of the plants, there are different types of chlorophyll. And you can separate out the chlorophyll types from a leaf by means of chromatography. The form in which we do it is very, very, very basic. You get specialized sheets containing like a silica gel which absorbs the solvent better, and then when it leaves the spot behind in the gel, you can scratch the gel off and you can wash the pigment out to get it separate. Okay? Genes. The diagram that they show with the little lines on them that represent genes. Have you seen this in the movies? It's done with electrophoresis. They do a chromatography using a potential difference across a sheet. And the different proteins travel up to different lengths. And then they can identify the genes. They can separate them out by means of a very specialized form of chromatography. All right. So chromatography separates substances on grounds of solubilities. If you look in your textbooks, there on page 30, they say estimating purity. Which substances are pure, A, B, C, or D? D. Why? Only contains one thing. Well, okay, then it's not pure. But we're assuming now they're all soluble in the same thing, same medium. Okay. They say, page 10, just below point 2, you will notice that some dyes rise up the paper more easily than others. What is the reason for that? Some are more soluble than others. Those that are more soluble will rise up the furthest. No. Okay. Then they say you can test to see whether it's pure or not. In other words, if there's only one substance that separates out of it, you can assume it's pure. If there's more than one, it is impure. In the second paragraph, I'm just reading, they say chromatography that uses paper is called paper chromatography. There are many other kinds of chromatography. One useful kind uses a column of pure sand in a glass tube instead of the paper, and the liquid runs down instead of up. This is called column chromatography. Okay, and then if it's got sand, if you have substances left at certain levels within that column, you can remove it and you can wash it from that substance to separate the substance out. Okay. Yeah, not at all. We'll just do very basic paper chromatography and the use of locating agents. Yeah, but they won't ask detailed anything on that. No, they can ask you on paper chromatography, determine the RA value, and if the spots are invisible, what do you do to see them? You treat it with a locating agent. You don't need to know specific locating agents at all. It's just very basic. Okay. They say chromatography can also tell us about the purity of colorless substances. Two common ways of doing this. Use a locating agent. Okay. This is a substance that is sprayed onto the paper after the experiment and which reacts with the colorless spots. How do you know when the experiment is done if you have got colorless spots? When the solvent reaches the top of the chromatogram, okay. Okay, yeah. Because some people don't think of it. Okay, done. That's it there. Yes. Yeah. Yeah. I think initially it's trial and error, but I think at this point they know if the substance you are testing for is a protein, they will use a certain locating agent for proteins. If it's sugars, they will use one. There are very specific locating agents. That's why I say you don't need to know the names or codes or whatever they use to do that, but it is very specific. Okay. If you've got something and you don't know whether your substances are only water soluble, you will repeat the process with water, with a solvent that will dissolve ionic substance, one that will dissolve covalent substance, or one that will dissolve sugars or proteins or carbohydrates or whatever. Okay. No, you don't use petrol. Petrol is molecular. Mm-hmm. The problem with the petrol is petrol itself is a mixture. So you must use a pure substance as the solvent. Okay? You would have to use one specific fraction containing whatever. Do you understand? Okay. All right. That then on chromatography, right at the bottom of the page, there they refer to electrophoresis. An interesting kind of chromatography is electrophoresis. An electric potential difference is made on the paper by attaching the top and the bottom to a direct DC voltage source. This creates a potential gradient, and different molecules will be pulled at different rates along this gradient. And that is how they use it to separate DNA during DNA analysis. Okay. Next heading there, compounds and elements. Definition of a compound. Two or more substances, chemically bonded. In terms of an element, it's a pure substance consisting of one kind of atom. Both compounds and elements are pure. Okay? If I have a mixture, what is the definition? Two or more substance? Not chemically bonded. No new properties. No energy uptake or give off. Yeah. Say again? Yes. Hydrochloric acid is actually a gas. And they dissolve it in water. Then that's why you get hydrogen chloride and you get hydrochloric acid. When I work with a gas that's not dissolved in water, okay, then I have hydrogen chloride. The moment I go and dissolve that gas in water, then I have hydrochloric acid. Technically, no. It's very soluble in water, so if it goes into the air or into a medium where there is water available, it's going to dissolve in the water and then it will be acidic. there's moisture in your lungs. Yes, then it acts as an acid. Okay. They dissolve in the rain. They ionize. Acids are molecular. They aren't ions. But when they react with the water, they form hydrogen ions and the corresponding negative, the chloride or the sulfate or the carbonate ion. Okay. I will get it out. I will find one. Okay. All right. So then there in your books on page 12, they simply refer to the second paragraph. They have a look at chemical reactions. Now, in general, your books refer to chemical reactions as being non-reversible, but this is not entirely true. Many chemical reactions are reversible, so I don't like that choice. So when you refer to a chemical reaction, then you refer to energy that is taken up or given off, and what is done with this energy? What is the energy used for when I want a reaction to occur? That activation energy.
It is used for bonding. Honey, nuclear.
Before bonds can form? This is very important, people. Remember the graph with the activation energy. Okay? If I have hydrogen, whether it's exo or endothermic, and I get water over here, what happens over there? Yes, I require energy to break those bonds. So I end up with hydrogen atoms and oxygen atoms. Now they can collide with each other, and when bonds are made, energy is given off. So we use energy during bond breaking. Okay, and energy is given off in bond making. And the reaction is exo or endothermic depending on how much energy is used for bond breaking and how much energy is given off during bond making. If bond breaking requires more energy than bond making gives, then it's endothermic. If bond making gives off more energy than what is required by bond breaking, then it is an exothermic reaction. All right. Nee, ek wag nie vir Albert nie. Hy's fine. When certain hydrocarbons, we refer to them as fuels. Most of the fuels we use are hydrocarbons. Now, the definition of a fuel is a store of chemical potential energy. And in general, we find that the fuels require less energy for bond breaking than what they give out during bond making, so they are exothermic. And we use this heat then to drive cars, to generate electricity, etc., cook food, and so on. Okay. Now, they say we can, when a fuel burns, most of our fuels are hydrocarbon fuels. What does this mean? They are made up out of hydrogens and carbons. So when they burn, the bonds are broken between the hydrogens and the carbons. They react with oxygen, so the products for the combustion of a hydrocarbon fuel is always water and carbon dioxide. All right. They summarize a couple of bulleted points there on page 12. They say all matter is made up of chemical elements. The elements are often joined chemically to form compounds. Most of the matter around us is made up out of a mixture of different compounds. For example, air is a mixture consisting of nitrogen, oxygen, carbon dioxide, water vapor, little bit of argon, helium, etc. Okay. Mixtures can be separated into pure substances. Remember, mixtures consist of elements. and elements, elements and compounds, compounds and compounds, eh? And they can be separated without having to break chemical bonds. So we say they can be separated by means of physical processes. Yes, that's like distillation or decanting or sorting. Yes, for a mixture. Okay. The next one, mixtures can be separated by physical techniques. We've just had that. And elements can be turned into compounds by chemical methods. When I turn an element into a compound, I'm going to make it react with something. So I'm going to break bonds and I'm going to make new bonds. So chemical reaction, then very simply making and breaking of chemical bonds. Then chemical changes, they have 3 bulleted points. A change in energy is involved. And then they've got the process is irreversible. Just put it in brackets. Sometimes it is, but it's not always. And the last one, the products are different from the substances that we started out with. They have new properties. So things like melting and boiling and evaporation and condensation are physical changes. We don't change the substance. But when we start cooking food or we start burning wood, then we get a chemical change. Because we start out with one thing and we end up with something completely different. All right. Kinetic theory, all matter, one of 3 states, solids, liquids, or gases. The phase change from a solid to a liquid is melting. A liquid to a gas is evaporation. A solid to a gas? Sublimation. And then it can go back. It can go from a gas. They can condense and form a liquid. And liquids can solidify and form solids. If a gas goes directly to a solid, we refer to it as sublimation. I see nowadays they put into the textbooks resublimation or deposition, just to distinguish between the 2 directions. No? Okay. Chapter 2. Let's just carry on with the stuff we've been doing. No, none of those tables are, you don't learn any of those things out of your head. Now, people, this whole first chapter is a little bit of, I don't think there's anything new except for the chromatography. It's just the differences between elements and compounds, pure and impure, mixtures and compounds or elements, etc. Now, in the next section, chapter 2, we are going to have a look at, well, basically, chemical bonding. Now, before we start off with bonding, they do the structure of the atom. What does an atom look like? Consists of a nucleus containing protons and neutrons. Okay? Put one there. And electrons. Now, technically, I don't really need that ring there. Why not? It's just particles. There isn't a ring around the nucleus as such. There's not a ring keeping the particles together. What keeps these particles together? Just simple attractive forces between... What will a positive and a neutral particle do? It's electrostatic attraction. Because positives attract neutrals. If I didn't have any neutrals, what would these positives do? No, I did Coulomb forces and van der Waals forces. If there were no neutrons in the nucleus, what will the positive particles do? They would repel. Would the nucleus be stable? No. So, yeah? Yes, but they do attract the electrons, but because the electrons don't form part of the central cluster. Oh, now I've got a wrong color over here. Let's do green. Okay, here are some neutrons. That is why the neutrons in the nucleus are there. It keeps the nucleus stable. What happens if a nucleus becomes too large? Like in uranium, when I shoot a neutron into it, they break up and we get nuclear reactions. But we are not busy with nuclear reactions now. So the chemical reactions that we work with do not involve the energy of the nucleus as such. It involves the energy with respect to the electrons. Now, electrons are negative, and you should know at this point that the negative electrons are found in shells around the nucleus. We have different shells. What's the difference between different shells?
The first one has 2 electrons and the rest is 8.
Yes, and what else? The first shell is the lowest energy. The second, so electrons with the least energy going to the first shell, next bit of energy going to the second shell, etc. Okay, now the electrons going into 28888 is not entirely true, but it's part of the model that we will work with, but there's a little extension on the higher level work where we go and have a look at the SPD distribution, but I'll explain that when we get there. For now, we are going to look at chemical bonds. So when one atom bonds chemically with another atom, the forces lie between the electrons, specifically on the outer shells. Chemical bonds involve outer shell electrons. And there are 2 basic ways in which we've done bonding. Which 2 bonds? Ionic bonding and Yes, we've done metallic and covalent bonding. And then we did metallic bonding, but metallic bonding is simple. It's just one metal ion with another metal ion and the delocalized electrons holding them together. All right. So, in general, when I burn a hydrocarbon fuel, I am breaking covalent bonds. All right. When I have an electrochemical cell, it's got to do with attractive forces between the ions that I can either break apart or put together. So chemical reactions involve outer shell electrons, the bonds between outer shell electrons. Okay. How big are atoms and molecules? What's the answer to that one? They aren't big, they're very small. Okay. People, if I have 2 grams of hydrogen, just 2 grams, have you got an idea of how small 2 grams are? I will have $23. I will have so many hydrogen molecules. So 2 grams, I mean, that's tiny. It's tiny amounts. But look how many molecules of hydrogen will I have. Okay? So these particles are very, very small. They are so small, they almost don't exist. Okay? They say there on page 70, it's an interesting part there in the Did You Know, they say if you throw a bucket full of water, now a bucket is probably about 5 liters, a bucket full or between 5 and 10 liters of water into the Atlantic Ocean in Argentina, okay, and then traveled across the Atlantic Ocean to Namibia and take out a bucket full of the Atlantic Ocean there will probably be some of the molecules from your Argentinian bucket.